Good evening. My name is Nicholas Hanley, and today I'm going to be discussing with you the chemical buffer systems against changes in hydrogen ion concentration that occur within our bodies. I have three objectives today. The first is to identify the sources from which the body receives hydrogen ions. Secondly, I'd like to list the three lines of defense that exist against changes in hydrogen ion concentration that are used in the body. And my third objective is to list and explain the four chemical buffer systems. That's where we'll focus a majority of this presentation. Okay, so first, let's talk about these sources of hydrogen ions in the body. If we look, um, most of these hydrogen ions are added due to the metabolic activities that occur within the body. So, um, main source there is going to be carbonic acid formation. Um, Again, all our cells as a metabolic waste produce carbon dioxide. This carbon dioxide could then combine with water that's readily found in our body and produce carbonic acid, as you can see in the center picture of the picture here. And that carbonic acid that can then dissociate into the bicarbonate ion and a hydrogen ion, which is what we use to determine pH and everything. So there's our source of that hydrogen ion. Um, the next thing is inorganic acids. Um, Basically, as you eat food in your diet, proteins found in there are high in quantities of sulfur and phosphorus. And by breaking down these components into or these substances into these components, sulfur and phosphorus, it can result in the production of sulfuric and phosphoric acid as byproducts, and both of those are pretty strong acids, so they'll dissociate into producing free hydrogen ions. You see it a lot with the grains and dairy products found in our diets. Um, it can kind of be balanced a little bit by the breakdowns of fruits and vegetables, which produce bases, so it neutralizes the acid when they're broken down. But the way our diets typically work as humans is we tend to intake a lot more things that produce acids than bases, so we get an overall um, production of some hydrogen ions from that. Finally, the third source of hydrogen ions in our body is via organic acids, um, the intermediary metabolism that occurs as we break down those. So, for example, fatty acids get produced during fat metabolism. Muscles produce lactic acid during periods of heavy exercise. And both of those, the fatty acids and the lactic acid, will partially dissociate and producing some free hydrogen ions. Again, this is a continuous process in our body. We're constantly making or generating these hydrogen ions. Um, there are certain diseases and disorders which may add hydrogen to the system beyond these sources. The example given in the book was diabetes mellitus. Um, that process, the breakdown of ketones, can produce keto acids, and diabetics that can lead to ketoacidosis and some other things. Okay, so those are the main sources of these hydrogen ions in our bodies. Okay, we have three main lines of defense against changes in pH or hydrogen ion concentration in our body. Um, Basically, what we want to do is we want to maintain the pH of this extracellular fluid in our body at around 7.4, a pH of 7.4. Even though 7 is neutral, that is slightly basic on the pH scale, but for our body, 7.4 is like the goal of where we want to be. So we want to balance the hydrogen ions that are formed to keep us at that 7.4 value. Um, Now, to do that, as we produce this free hydrogen ions, we need to be able to remove them and eliminate them from our system. Um, so we do that in three main ways. We can use chemical buffering systems, we can use a respiratory mechanism of pH control, and we can use a renal mechanism of pH control. Um, for the rest of this presentation, we're going to focus on these chemical buffer systems. Um, the chemical buffer system basically doesn't eliminate the hydrogen ions. It's going to kind of mask it or hide it by binding the free hydrogen ion with another chemical to kind of mask it as it's traveling through the blood. If it's not free as a hydrogen ion, it cannot affect the acidity. Um, the respiratory and the renal mechanisms are actually going to actually remove or eliminate the hydrogen ions from the body, from the system. All right, so let's go and look at those more closely. Okay, chemical buffer systems. In a chemical buffer system, the chemicals involved can either bind to or release free hydrogen, and that allows us to either increase the pH or decrease the pH depending on what's going on in the body. The whole purpose here is we want to maintain our pH level. So in all buffer systems, it's going to be a mixture of two chemical substances. One of those substances will be able to bind with free hydrogen ions. The other is going to be able to produce free hydrogen ions. So in most cases, or in all the cases here, these two chemicals are going to exist in a reversible chemical reaction. The picture here is just, again, on the last slide we talked about how we want to maintain pH. 7.4 is like the average value we're looking at. As long as we can keep it into this normal pH range here, between 7.35 and 7.45, that's where we want to be. When we start getting outside of that range, if we drop below 7.35, the acid side of this, we have acidosis. And if it drops too far, as we can see, we go from a disorder to something our body can't compensate with, and resulting in death. Similar thing happens on the other side. If we get too basic or too alkaline, if we get above 7.45, we go into alkalosis. And again, if we keep rising there, we could end up with death, and that's not something we want to do. So chemical buffer systems, we want to maintain that pH, and we're going to do what we can to do that. So if we look, there are four main buffer systems in our body. Table here is from our book. It does a good job of explaining the major functions, but we have four. Okay, so the carbonic acid bicarbonate buffer system, the protein buffer system, hemoglobin, and phosphate. We're going to look at each of those, four of those, as we go through this next. Okay, so in the carbonic acid bicarbonate buffer system, we're going to follow this equation here. I left part of it out. Carbon dioxide and water are going to combine to form carbonic acid. But that carbonic acid can then dissociate into hydrogen ions in the bicarbonate ion. That is kind of the basis here. The carbonic acid and the bicarbonate are the two things we're going to focus on. So basically, if we have carbon dioxide, it's going to go there and kind of react with the water to make that carbonic acid. So for this one, this is the primary buffer of extracellular fluid. The carbonic acid and bicarbonate are very abundant here, so we're able to use it and do what we need to. It's very effective for two main reasons. Again, like I just mentioned, it's available. There's an abundance of them there, so it's there and it can work, and we can do what we need to maintain that pH. We can release hydrogen ions, we can bind with them, whatever is needed there. And then both of those components, both the bicarbonate ion and the carbonic acid, are highly regulated by the body. So we can keep track of those, we know they're there, and we can use them. The bicarbonate ion gets regulated by the kidney, and the carbon dioxide and carbonic acid gets regulated by the lungs. So what's interesting here, if carbon dioxide levels and thus carbonic acid increases, it's going to shift this reaction to the left, to the right here, to the forward reaction, producing more hydrogen ions and bicarbonate ions. If bicarbonate increases or hydrogen ions decrease, then we're going to work the other way. So we can kind of adjust this as needed depending on what's going on in the body. So basically, when we add new hydrogen ions to the plasma from any source other than carbon dioxide, because if you look at the last bullet here, this buffer system cannot work on itself. Okay, so buffer changes as a result of a bunch of other acids, but it cannot buffer changes in pH due to carbonic acid. Just can't work on its own. So when new hydrogen is added, um, the preceding reaction is driven toward the left side of the equation. So we're going to go more this way. The extra hydrogen ions are going to bind with the bicarbonate that's there, and that's going to kind of pull those hydrogen ions out of solution and maintain the pH where we want to go. Um, if the hydrogen ion concentration was to fall below normal for some reason, again, other than changes in carbon dioxide loss, then the reaction is driven towards the other direction. We're going to move towards the right, and we're going to generate additional hydrogen ions by the breakdown of the carbonic acid. So both things are here, and we can work with what we need to. Again, it can't work on itself because if we're producing hydrogen ions from an increase in carbon dioxide, well, an increase in carbon dioxide will shift the reaction this way and make more hydrogen ions, and it doesn't work out. We're not going to ever be able to swing it back the direction we want to go because we're always forcing the reaction in the direction. If we look at the protein buffer system, protein buffering system is more important intracellularly. Now, the bicarbonate buffer system, that was more extracellular. We're going to use this one more intracellular. And the main reason that is the most plentiful buffers of body fluids are the proteins, including intracellular and the plasma proteins. The reason they're such good or effective buffers are due to their amino acids. The proteins are made up of many amino acids linked together in sequence. Some of the amino acids we use to build proteins can be acidic, and some are basic. Okay, the basic amino acids have the ability to bind to a hydrogen ion that's free, whereas the acidic amino acids have the ability to release a hydrogen ion when necessary. So just like the bicarbonate and the hydrogen ion and carbonic acid was able to bind to or release hydrogen ions, we can do the same thing with the various amino acids in the protein. Again, more effective intracellularly because of the more abundance of proteins there. In the extracellular fluid, bicarbonate and bicarbonic acid are more prominent, so they're going to be used. Okay, hemoglobin can also serve as a buffer. We talked about how all metabolic reactions produce carbon dioxide. Well, once they produce that, the carbon dioxide enters the blood and eventually travels to lungs to be released. That carbon dioxide is going to get converted to hydrogen ions and bicarbonate ion within the red blood cells as a result of the carbonic anhydrase. So carbon dioxide and water react to form carbonic acid. That carbonic acid then dissociates into the hydrogen ion and bicarbonate ion. So this free hydrogen ions here would cause the blood in these venous systems, bringing the blood back to the body, would cause it to rise or increase in pH. And, we don't want our venous blood to be higher in acidity than our arterial blood. There is a slight increase in venous blood, but what happens is that hemoglobin molecule that's free from the oxygen will bind with that free hydrogen ions, and that will, if the hydrogen ions aren't in the blood, aren't in solution, they can't contribute to the pH, so that maintains the pH throughout that system. Again, once we get to the lungs, these reactions are going to reverse. Carbon dioxide is going to be produced again and gets released into the environment. Our final buffering system is the phosphate buffer. First, this is pretty important in the kidneys, the urinary system. There are two components to this buffer system. The acid phosphate salt, which is the first one, and this one is able to produce hydrogen ions when in solution and when they're needed. And then we have the basic phosphate salt here, which can accept or bind to hydrogen ions when needed. So basic brings the hydrogen in, binds to it. The acidic one is going to release the hydrogen ions. So we have our two components of our buffer system. If we need more hydrogen ions, then we're going to shift this way. If we need more, this reaction will shift this way if we need more hydrogen ions. If we have too many hydrogen ions, then they'll combine with the basic salt and produce this way. So we can go back and forth on that one as well. Basically, the hydrogen and the sodium, we're going to switch a hydrogen ion out for a sodium ion in the compound to make things work the way we want. So it is a good buffer. Its concentration in the extracellular fluid is pretty low, so it's not very important as an extracellular fluid buffer, but definitely effective within the urinary buffer, because phosphates are most abundant within the cells. This system contributes to that intracellular buffering. Again, the only thing that works more effectively intracellularly is the protein buffering system. So this is second to proteins. But the reason this one works, we normally consume more phosphates than we need to. So the excess phosphate gets filtered through the kidneys and it's not reabsorbed. And so it remains in the tubular fluid to become urine and excreted within the urine. That excreted phosphate that's in there buffers the urine as it forms because it removes the hydrogen ions from solution. So no other body fluid buffer systems are present in the tubular fluid to play this role. So this is the main or only thing that is able to buffer urine formation. And then finally, these chemical buffer systems are going to serve as our first line of defense to maintaining our hydrogen ion concentration. Again, the main reason here, it's the first mechanism to respond. It's quick to react to those changes in hydrogen ion concentration because the molecules are there. Because it's a buffer system, both things are present. We can either add hydrogen ions or we can remove them immediately as needed. So that quick response is very vital, and that's why that's the first line of defense. Again, what we're doing here is we're trying to hide this hydrogen ion concentration or hydrogen ions that are free until they can be eliminated. If they're not dissolved in solution, they cannot contribute to the acidity, therefore the pH. We can maintain the level we're at. The chemical buffer systems are able to hide the hydrogen ions, but they don't eliminate them. The respiratory and renal mechanisms that we talked about at the beginning and need to be expanded more, but not in this presentation, are the two systems that are going to eliminate those hydrogen ions. Again, to look at the objectives, we identified the source from which the body receives hydrogen ions. We listed the 3 lines of defense against changes in hydrogen ions used in the body. And then we listed and explained the 4 chemical buffer systems. Here are my references, and thank you for your time.